Hello, good evening. Today we're gonna deal about bacterial capsule. So first of all, what is the bacterial capsule? So bacterial capsule is a anatomical structure especially designed by bacteria or any organism to adjust in adverse condition. And to aid in virulence of the pathogen of the organism. So typically the bacteria capsulated bacteria are more pathogenic than non-capsulated bacteria. So capsulated bacteria always has K antigen. So what is the bacteria? What is the capsule? Capsule is an anatomical structure designed by the bacteria or any organism to adjust in adverse condition and to aid in a virulence of the organism. So capsulated bacteria are more pathogenic than non-capsulated, and capsulated bacteria always have K antigen. So talking about the location where it lies in a cell. See, it is always lies outside of cell. When you draw the structure of bacteria, so first of all, we can draw the cytoplasm with cell membrane. It's a cell membrane. Here is cell organelles, ribosome, 70S ribosome. Bacteria always contain 70S ribosome. It's a protein factory. It is a circular plasmid and DNA, RNA. So above the cell membrane, there is a peptidoglycan layer. In case of gram-negative bacteria, there is also an outer membrane. Gram-negative bacteria, there is also outer membrane. It's a lipopolysaccharide. And now some capsulated bacteria, the capsulated bacteria has a layer of capsule which lies outside the cell. So what is the function? What is the function of capsule? What is the significance of capsule? So, significance or function of capsule. First things, it is a anti-phagocytic. It means that when phagocytic cell engulf the capsulated bacteria, capsule prevents from a phagocytosis mechanism and permit the bacteria to survive inside the phagocytic cell. Point number 2, it also helps in attachment. So attachment is the second steps of host parasite interaction. So it helps in attachment to the host cell. Point number 3, it also helps in formation of biomolecule. Formation of biomolecules. Formation of biofilms. Point number 4, it is antigenic in nature. Antigenic means when it is inserted in a body, it can generate the immune response in our host, and it's also a virulence factor. So they are the overall functions. So main function is a antiphagocytic in nature. Now talking about the biochemical nature or chemical nature of capsule. So what is the biochemical nature or chemical nature of capsule? So capsule typically has two types. One is a polysaccharide capsule. Polysaccharide capsule. It means that the capsule is made from a carbohydrate. So polysaccharide capsule usually soluble in water. And such types of capsule helps in formation of sticky. So capsulated bacteria have a colony polysaccharide capsulated bacteria have colony nature, is usually sticky, liquid, and smooth in nature. So they are the features of the capsulated bacteria. So some of the bacteria which can form the polysaccharide capsules like Klebsiella pneumoniae, likewise Streptococcus pneumoniae, so on. Second one is a polypeptide capsule. It means that the capsule is protein in nature. So example, Bacillus anthracis. So its capsule is a polypeptide and it is a polymer of D-glutamic acid. So the capsulated capsule has a chemical nature, either by polysaccharide in nature or polypeptide in nature. Now, talking about the diagnosis and detection of the capsule in a bacteria, how it can be diagnosed, how it can be detected. So, most common usual method is a very abstract. So, detection techniques of capsule. First, most common usual method is negative staining. So, negative staining is a procedure in which we use a negative stain. In process of negative stain, background is a dark and capsule is a clear halo zone. So, most commonly use a stain in a negative stain, India ink or negrosine. Point number 2, Anthony stain. See, the other staining techniques which also helps in detection of capsule. So in this staining we use positive stain, usually crystal violet. It's a positive stain. It stains the cell as a violet color, and copper sulfate as decolorizer. So cell takes the color of crystal violet and capsule is still as a clear halogens. Point number 3, Quellen reaction. So it is antibody induced capsular swelling reaction. So it is also capsule detection test. Point number 4, latex agglutination test. So it is a rapid diagnostic test. It's a rapid diagnostic test of capsule. So they are the procedure, method or techniques that we use for the detection of capsule in a bacteria. Most common or usual method is the negative stain, another is the Anthony stain, and quelling reaction and latex agglutination. Now, the bacteria which can form the capsule, also the bacteria which contain the capsule. So, very common mnemonics for the remembrance of the capsulated bacteria is the mnemonics. So we can remember the bacteria which forms the capsule. Some killer bacteria have pretty nice capsule. So this is the mnemonics, a common mnemonics. Some killer bacteria have pretty nice capsule. What S stand for Streptococcus pneumoniae. So we can call it the pneumococci also, pneumococci, or gram-positive diplococci at 180 degree, or we also call smallest chain cocci bacteria. smallest chain, cocci, bacteria. So they are the common names of the Staphylococcus pneumoniae, and it is the most common child pneumonia in children. So likewise, S stand for Staphylococcus mutant is a causative agent of dental caries. So on Staphylococcus agalactiae. So they are the bacteria which can form the capsule in polysaccharide in nature. So on K represent for Capsule pneumoniae. It is also gram negative. It is a gram negative bacteria. It usually causes pneumonia and UTI, urinary tract infection. Third one, B represent for Bacillus anthracis. So its capsule is a polypeptide and polymer of the glutamic acid. So what is the causal? It causes the anthrax. The anthrax is also called Wool-Sorter's disease. Wool-Sorter's disease. So the anthrax is commonly associated with the person which are close contact to the animals. For example, farmers, butchers, veterinarians, hide seekers, or wool sorters. So they are the zoonotic disease. Anthrax is a zoonotic disease. It means that it can transmit from animal to man. So likewise, H stands for Haemophilus influenza. So Haemophilus influenza is a causal agent of meningitis. It's also gram negative, and otitis media. P represents for Pastorella, Maltophila. So this is also bacteria gram negative. It is called animal-induced pneumonia. It's also zoonotic. And N represents for Neisseria meningitis. So it's a causal agent of meningitis. It is a gram negative diplococci at 90 degree. So last one, C represent for Cryptococcus neoformans. It's a fungi. So all of the organism here deals Staphylococcus pneumoniae, mutans, agalactiae, Capsule, Bacillus, Haemophilus, Pastorella, Neisseria. All are bacteria except Cryptococcus neoformans. And all the organism given here have a polysaccharide capsule except Bacillus anthracis. Its capsule is a polypeptide and polymer of D-glutamic acid. So we deal here the bacteria or organism containing capsule with mnemonics. Some killer bacteria have a pretty nice capsule. Now, talking about the MCQs, what are the possible MCQs related to the capsule? So some common questions which are asked in a question, competitive examination. Question number 1. The most common method of capsule detection. So point number 1, A, negative staining. Point number B, Anthony staining. C, Quellen reaction. D, latex agglutination. So we can treat negative staining. Question number 2. The rapid diagnostic test of capsule staining is option number A, negative staining. Option number B, Anthony staining. Rapid diagnostic test for the capsule is point number C, Quellen reaction. Point number D, latex agglutination. So we can take latex agglutination. Likewise, the question number 3, antibody induced capsular swelling reaction is. So option number A, Quellen reaction. Option number B, agglutination reaction. Option number C, ASO test. Option number D, negative staining. So we can treat Quellen reaction. Similarly, question number 4, capsulated bacteria have always antigen. M antigen, K antigen, O antigen, MTR antigen. So always we take the K antigen. So other questions. Question number 5. Which is not the function of capsule? Which is not the function of capsule? Option number A, so virulence factor. Yes, capsule is also virulence factor because it permits the survival inside the phagocytic cell. Antigenic in nature. C, helps in biofilm formation. Yes, and option number D, motility function. So all of the options here given are function of capsule except motility. Motility is the function of flagella. Question number 6. Capsule always lies cell. As a lies. Point number 8. Between cell membrane and cell wall. B. Between peptidoglycan and outer membrane. C. Lies outside the cell. D. In cytoplasm. So we always take lies outside the cell. So this is the MCQs that are related to capsule. Question number 7. Among the flowing option given below, which one has polypeptide capsule? So option number A, Streptococcus mutant. B. Bacillus anthracis. D. C. Klebsiella pneumoniae. Option number D. Haemophilus influenza. So Bacillus anthracis has polypeptide capsule. So this is the questions we deal related to the capsule. So hope you guys you all understand these MCQs and their chemical nature and function of capsule. Thank you very much.
के तपाईहरु लाइसेन्स एक्जाम क्र्याक गर्नको लागि राम्रो स्टडी सेन्टरको खोजीमा हुनुहुन्छ
भने आउनुस् तपाईहरु पनि जेनिक स्टडी सेन्टर